like 8 weeks as every other scholar. She's a junior at Duke University. She has some lab experience, but I can say that she was from the beginning looking very confident and start working, and she was very, very efficient. I'm so happy having her here. She helped me a lot. I hope you learn a lot, and I have to say that I learned a lot also from you. So she worked on identification and expression analysis of ammonia transported Setaria Viridis, and she has done a great work. So let's see her. Thank you.
So thank you, Leah, for the introduction. So my project this summer has contributed to a larger collaboration between 2 labs in the UK and 4 in the US, one of which is Dr. Viridis here at the Nobel Foundation. And the goal of their work is to engineer a symbiotic relationship between non-leguminous crop plants and nitrogen-fixing bacteria in order to put more nitrogen into the soil without relying too heavily on fertilizers. And so the outcomes of this would hopefully be to both increase crop yields in poorer areas of the world where they can't afford as much fertilizer, but also to reduce the amount of fertilizer needed for farmers in more prosperous areas to maintain their already high crop yields. And some of the reasons we want to turn away from using fertilizer are that they're 1, expensive, 2, wasteful, since farmers have to apply more nutrients to the soil than the plants are able to uptake and use. They're also bad for the environment since runoff into rivers and groundwater causes pollution. Now the reason we need fertilizer in the first place is to provide the plant with nutrients that it needs to get from the soil. And one of the most important is nitrogen, since it's a component of the major macromolecules. Unfortunately, however, 99% of the nitrogen on the planet are available in this form. So since the 2 nitrogens are held together by a very stable triple bond, most organisms haven't evolved an enzyme capable of breaking it down into a usable form. The ones that have are called diazotrophs, and they're microorganisms. Many of them live in the soil. And they have an enzyme called nitrogenase that can break down the ammonia, the atmospheric gas, and incorporate it into a fixed form, ammonia. And so the way plants uptake their nitrogen naturally is in these 2 major forms, ammonium and nitrate. And so those are put into the soil via different mechanisms. A lot of organisms live in the soil, or nitrate can also get in the soil from a reaction driven by the energy from lightning. And then there's a family of plants called legumes that have evolved the ability to form these root nodules and shelter rhizobia, a type of nitrogen-fixing bacteria. And by sheltering them from oxygen, the nitrogenase enzyme can work at a better capacity. And so in exchange for this place to live and nutrients, the rhizobia gives the legume nitrogen in the form of ammonia. And so if we're looking to take the system and put it into other plants, there's a lot of aspects to pay attention to, both from the side of the microbe and from the side of the plant. And the side my project dealt with was the plant taking up ammonium. And we looked at how the plant did this in a natural situation to see if anything would need to be adjusted in a situation where it was directly linked to a microbe. We're also interested in studying ammonium because it takes less energy for the plant to incorporate it into its biochemical pathways and to turn it into amino acids, since ammonium can directly enter this reaction here, whereas nitrate has to first go through 2 reactive steps before becoming ammonia. So the transporters responsible for bringing ammonium across cell membranes are called the AMT-MEP rhesus family, and these are found in organisms across different domains and kingdoms, but the ones in plants are called AMTs. And so in a given plant genome, many AMTs are expressed, and some of them have redundant functions, some of them don't function at all, but then sometimes the plant upregulates certain ammonium transporters depending on the environmental situation. So for example, there's high affinity and low affinity ammonium transporters. And the reason they're kept in this balance is because although a certain amount of ammonium is necessary for the cell to survive, too much can be toxic. So in a situation where there's not a lot of ammonium in the environment, the plant would want to upregulate the more high affinity ammonium transporters to grab as much ammonium as it can and bring it into the cell. Whereas in a situation where there's a lot of ammonium in the environment, the cell would want to express more low affinity ammonium transporters so that it allows in ammonium at a more manageable rate. And so the two questions, so here are the two questions that my project was going after, and I'll first start by talking about the first one, which is what ammonium transporters are coded for in the Setaria genome. And the reason we're looking at Setaria as a model organism is because it has a short life cycle and a sequenced genome, but it's also representative of C4 grasses like maize and switchgrass, which are important to agriculture. And so we started by looking at proteins that were already characterized in Arabidopsis in this database, and we looked for homologs using phytosome in the Setaria genome. We used interposed-scan to find motifs in the genes we found to make sure that they had ammonium transporter motifs, and then TMHMM to find transmembrane passes. And so these were the 8 that we found. 2 were type 1 and 6 were type 2, and we'll talk a little bit about what that means later. The black up above represents the peptide link, the yellow is the ammonium transporter motif, and then the red are the transmembrane passes. And looking more closely at that, the way we found the transmembrane passes was we put our peptide sequence into TMHMM, and it compared our sequence to sequences of characterized proteins, and gave us this graph of the probability of a particular segment being inside the cell, outside the cell, or in the transmembrane region. And so I just took a graph like this and made like a schematic of what this graph might look like embedded in the cell membrane. And so we did this for all 8 of our genes, and we found a major difference between our what we call our type 1 and our type 2. Type 2 had this longer extracellular region in the middle, and that could say something interesting about its function. So it could be a place for the substrate to bind, it could be a signaling platform, and we don't know, but it's interesting. So then we did an alignment of our 8 peptide sequences to see how similar they were. And we found that the 2 type 1s had an 83.6% similarity, so they were pretty close. Whereas our type 2 had a range between 53.6 to 82.3, so not as close. And then between the 2 types, it was only around 20%. And so we put that into a phylogenetic tree with characterized proteins from other organisms, and we found where they fit, and so our type 1s fit into this group with other type 1s, and our type 2 in this other group. And our final in silico test was to run our sequences through the substrate specificity prediction to see what kind of substrates it would be able to transport. And we did this because we wanted to make sure that it would make sense for this protein to transport ammonium. And since all of them could transport cations, we decided to proceed with these 8. And so that brings us to the second question, which is how are the expression levels of the AMTs, and how are their physiological traits affected by treatments under varying nitrogen sources, under varying ammonium concentrations, and then stress followed by resupply. And so we set up to grow these plants. And so we watered them all with a Hogan solution that contained all the nutrients we needed except for except for nitrogen. And so they all had a consistent amount of other nutrients. And then we gave them the type and concentrations of nitrogen we wanted. And so for the first part of our question, the source of nitrogen, we had 5 millimolar just nitrate, and then we had 2.5 millimolar ammonium nitrate to keep it at 5 millimolars of nitrogen. We also had an experimental setup for the asking about the concentration of ammonium, and so we had ammonium chloride going from 0 millimolar to 5 millimolar. And then we also had a condition with full nitrogen, and that meant it had 5 millimolar of nitrate and 0.5 millimolar of ammonium, because this is what we would normally feed them. And so we grew them like that for 10 days, starved them of any nitrogen for 7 days, and then harvested tissue after resupplying them with nitrogen after these time points. And so we took physiological measurements at 10 days and 30 days and found a lot of variability after 10 days, and so we're just presenting the ones for 30 days. And this was to see how the plants did overall. And we found this trend pretty consistently with all our measurements, where the higher the concentration of ammonium, the better the plants grew. And so it's kind of interesting because when you have ammonium nitrate, it grows almost twice as well as the only nitrate condition, even though they have the same amount of nitrogen. We see this same trend with our dry weights and our fresh weights for both root and shoot. And then for our SPAD measurement, which measures the plant's photosynthetic capabilities, we kind of see a more consistent SPAD reading for the different sources of nitrogen, but there's a little bit of an increase in SPAD when you get to 5 millimolar. And then we couldn't take the measurement on our 0 millimolar ammonium condition because the leaves were too small. For leaf area, it followed the same trend as before. So then we get into our expression analysis, and we did a qPCR. So first we harvested our tissues at the times we wanted. We isolated RNA and then treated them with DNase in order to clean up any DNA that was left behind after the isolation. And then we took reverse transcriptase and made cDNA and ran the qPCR with these two reference genes and their ubiquitin and elongation factor, which are important to the cell survival, so we knew they would be in the tissue, and we used them to compare levels. And so this is what we found. So for AMT1A, which was one of our 8 genes we looked at, it's kind of interesting because once the plant has enough nitrogen, so at like the 2 millimolar and above concentration, and also in the ammonium nitrate and only nitrate conditions, it had pretty similar expression levels of this gene. However, when there was a lower amount of ammonium given to it, like it was overexpressed. And it's also expressed in the shoot and root. We just wanted to make sure see where it was expressed. And then when we did the starvation and resupply, we found that before the resupply, they were expressed a certain amount, but that increased after it was resupplied with ammonium. And then we see a similar trend in 1B, except this time it's not expressed in the shoot. And this trend is the same as well as with 2B, but a comment here is that this is a much smaller scale. We're looking at the differences between the bars though, and it follows a similar trend, although it's not expressed very highly at all in these conditions. And same with here, although it's expressed a lot under these normal conditions, it's also expressed in shoot. And then going on with 2D, similar results. For 2E, it kind of almost follows an opposite trend where, and we don't see as much differences between them. 2F, it's expressed slightly more in the 5 millimolar concentration. It's highly expressed in the ammonium nitrate condition and low expressed in only nitrate, and it's expressed in the shoot somewhat. And so the conclusions we drew from this was that type 1 AMTs are expressed at higher levels than type 2 AMTs, looking at the scales that the graphs were on. These genes were expressed in both shoot and root, whereas 1B and 2D were only in the roots. All AMTs were induced at 0 and 0.1 millimolar ammonium, and then the 1A and 1B expression didn't change much when there was enough nitrogen for them. 2B and 2D had relatively low expression levels when the nitrogen availability was high, and then 2E and 2F increased at 4 hours after resupply. And so in the future, we would want to test the functionality of these AMTs, and we would do that with yeast mutant models, and then we would want to find the tissue localization of the AMTs, find out which membranes the transporters are located on, and then test expression levels of glutamate synthase, since that's the next step in the pathway to incorporate the ammonium into the cell. And so it would be interesting to see if that was upregulated when the transporters were. And so with that, I'd like to thank everyone at the Viridis lab for welcoming me in from the beginning, especially Yoon for helping us with the leaf area measurements, Yvonne for helping with the RNA isolation and the qPCR, and Rita for helping us harvest tissues. I'd like to thank the greenhouse staff for taking care of our plants, and then Frank and Emily for checking in on us every now and then and taking care of us, and Adam for doing this presentation. So that, any questions?